Hello and welcome to SAM online class. This is Dr. Subrat Kumar Mahanti. Friends, today we shall discuss about riboswitches. Riboswitches are alternative forms of mRNA secondary structure. These are the mRNA sequences or elements which are present in the 5' untranslated region of coding sequence. The 5' untranslated region of coding sequences contain these mRNA elements. The riboswitches bind with small ligand. The binding of riboswitches with small ligand induce conformational change in one domain. The conformational change in one domain induce conformational change in another domain. This conformational change leads to the formation of a terminator structure that directly interfaces with the transcriptional machinery and shuts it down. All riboswitches possess metabolite sensing RNA sequence. This metabolite sensing RNA sequence, which are present in all riboswitches, can adopt one of the two secondary structures. These secondary structures allow the transcription to proceed or not to proceed, and the decision depends on a molecular substrate of the cell whose existence is influenced by the gene that is encoded by the RNA. Riboswitches recognize various ligands including amino acids, purines, vitamins, cofactors, amino sugars, metal ions and other. Riboswitches are widespread in bacteria. In Bacillus subtilis, 5% of the genes are regulated by riboswitches. These mRNA sequences are also present in archaea, fungi, in plants and in animals as well. These riboswitches contain two important domains. One domain that binds with ligand is called as aptamer. Aptamer is the domain that binds with specific ligand. Other domain is expression domain or expression platform. The expression domain is capable of forming a terminator structure that causes termination in transcription. This is a riboswitch in the absence of ligand. This diagram is a 5' untranslated region in the absence of ligand. In the absence of ligand, this 5' untranslated region is transcribed by the enzyme RNA polymerase. In the absence of specific ligand, this aptamer domain is transcribed and the expression domain adopts an anti-terminator structure. After the formation of anti-terminator structure, transcription proceeds through the expression domain into the coding sequence. So the gene is transcribed. When the ligand is absent, this 5' untranslated region assumes a default conformation. But in the presence of ligand, the binding of ligand induces a conformational change in the aptamer domain. You can compare the conformation of the aptamer domain before the binding with ligand and after binding with ligand. After binding with specific ligand, there is specific conformational change in the aptamer domain, which also induces another conformational change in the expression domain. The conformational change in the expression domain causes the formation of a terminator structure. The formation of this terminator structure effectively block the enzyme RNA polymerase and the transcription is prevented. This description of riboswitches fits into a majority of cases, but there are two variations from this model. First is, it is possible for the default position of a riboswitch to be present in the terminator conformation. For example, the 5' untranslated region represents the gene that code for the enzyme amino-acyl tRNA synthetase. The enzyme amino-acyl tRNA synthetase catalyzes the binding of amino acid with their specific tRNA. When amino acids are absent, the tRNAs remain in uncharged condition. These uncharged tRNAs bind with the ligand binding site of the aptamer domain, and this induces the formation of this anti-terminator conformation. The formation of anti-terminator conformation allow the transcription and subsequently the translation, so it results in the synthesis of the enzyme amino-acyl tRNA synthetase. The situation becomes entirely different when charged tRNAs are present but uncharged tRNAs are absent. In this case, the default position of the 5' untranslated region causes the formation of a terminator structure. The formation of terminator structure ultimately terminates the process of transcription. Finally, there are some bacterial bioswitches in which, like attenuation of tryptophan operin, the alternative conformation of the 5' untranslated region causes the transcription to be completed. But when the ligand binds to the aptamer domain, this induces a conformational changes that inhibit the process of translation. Friends, this is about bioswitches. Please share and like this video, and also subscribe my YouTube channel Sam Online Class. Thank you very much. Good night. Bye bye.